And our next presenter will be Dr. Nalasivam Palisamy from the Henry Ford Health Institute. And he has an excellent presentation ahead of us, combined immunohistochemistry and RNA-ish to elucidate tumor molecular heterogeneity in prostate cancer. Thank you very much.
Hi everyone, I'm Nala Palaniswamy. Today I will be talking to you about the application of combined immunohistochemistry and RNA in situ hybridization to study the molecular tumor heterogeneity in prostate cancer. So in this presentation, I am not going to overwhelm with any data. So what I'm trying to do is I will just share my experience because I'm one of the early adopters of this technology, both RNA-ish and combined IHC and RNA-ish. So I would like to share my personal experience in setting up this technology in my lab. Then I will show you how successfully we have used this approach to understand the molecular heterogeneity of prostate cancer. The outline, I don't have any disclosure to do, so I'm not working for ACD Bio or Biotechne, so I don't have any personal disclosures or conflict of interest. So the outline of my talk is going to be, first I will show you how comparable the RNA in situ hybridization with the traditional immunohistochemistry, because I think this will be helpful for any new lab that they are trying to set up this RNA in situ hybridization technology, because my actually aim is to actually share my experience to the laboratory people who are interested in actually setting up this technology for the first time in the lab. And also, I'm sure it will be useful for some people who have already have been using this technology. So whatever that I'm presenting in this lecture is going to be not restricted only to prostate cancer, since I have been kind of primarily working on prostate cancer, but it is quite adaptable to other solid cancer or any type of cancer as well. So initially, first I will show you how comparable the RNA in situ hybridization technology is comparable with the traditional immunohistochemistry. Then I will show you how we can actually use the RNA in situ hybridization to detect some of the AIDS gene fusions in prostate cancer, because we have to use only RNA-ish, because for some of the genes we don't have any good antibody. So we were the one first, we used the RNA in situ hybridization for detecting this gene fusion prostate cancer. So I will show you some of the publications as well. Then using this combined approach, I will show you how we identified distinct molecular subsets of prostate cancer. Then by successfully using this approach, we used this technique to study the tumor molecular heterogeneity in prostate cancer by preserving the cell morphology, by doing the in situ analysis. So like traditionally people do next-gen sequencing by taking samples from different areas of the tumor, then do the sequencing, they look for copy number changes, mutation, other things. But this is a powerful approach where you are not biased with the tumor selection. You will be able to analyze the tumor as it is. So I will show you some good examples how we successfully use this. Then since the RNA in situ hybridization is a powerful approach, this will be very useful for the detection of non-coding RNAs for in situ analysis, because non-coding RNAs, obviously you cannot develop any antibodies. So I think for in situ analysis of non-coding RNA, I think RNA is a very powerful approach. So I will show you some examples how we use that. And then at the end, I will quickly show you, you can also use the DNA fish, that is a traditional two-color or single-color fish technology that can be combined with RNA-ish, so that you will be able to detect both the DNA copy number and also the RNA expression simultaneously for the gene of interest. Because sometimes you will see the gene amplification, but the question will come, even though that gene is amplified at the DNA level, is it really expressed at the RNA level? So if you have that type of question, you will be able to answer that question simultaneously by doing the DNA efficient RNA-ish. So with that, in this slide, I want to actually give some tips based on our experience. I thought this will be very useful for the people who are trying to establish this technology for the first time in your lab. So this is mainly for combining both IHC and RNA-ish. So particularly when you are planning to do RNA-ish, make sure you use freshly cut tissue sections for FFPE. It is okay to have the blocks as it is, but when you cut the slides, make sure it is fresh, and then even if you cut before, make sure they are stored at a good temperature like -20 or -80. And if you have already, if someone is providing you the already cut slides, make sure that they are not older than 3 months. Sometimes, even if they are older than 3 months, you will see the signals, it may be working, but the quantity and the quality may be compromised. You may not see the actual tissue level expression, so make sure that you use the fresh slides. Then before doing the IHC and RNA-ish, make sure that you use the positive control probes to make sure the RNA integrity is preserved. I think there are some good positive control probes that are available. Then the other important point is people may be wondering, because if you are trying to use it in the clinical lab, both manual and automated methods can be used for doing the RNA in situ hybridization. So I think the automated methods like I think DACO or the Ventana systems can be used for the automated analysis, but the manual procedure also working equally well. Then other important point that I would like to mention is if you are doing the multiplex assay, make sure you want to test each probe individually first, make sure they are working before engaging the dual approach. And other important point is when you are doing both IHC and RNA-ish on the same slide, make sure you do the RNA-ish first before doing the immunohistochemistry. But you cannot do the other way around. Don't do IHC first, then RNA-ish, because that will kill all the RNA. You may not see anything like that. But one good advantage is if you do the RNA-ish, there is no need for doing the secondary pretreatment and antigen retrieval and all those things. Whatever the pretreatment that we are doing for RNA-ish, that is more than sufficient for the immunohistochemistry. And like I mentioned before, DNA-fish and RNA-ish can be performed on the same tissue. The other important point is it is important when you are doing both IHC and RNA-ish, you have to have some prior knowledge about the expression pattern of the marker, because some may be nuclear, some are cytoplasmic, or some are membrane. Make sure that if you are doing two markers, say like two markers for IHC, if both are expressed in the nuclear, you may not be able to solve it. So make sure like one is cytoplasmic or membrane, the other one is nuclear, like that, so that you will be able to distinguish the expression pattern. I think these are, I thought these are all some of the important key things that you want to keep in mind before setting up the experiments. And if you have any other technical issues, feel free to contact me or the ACD Biotechnical Support. They have been excellent in troubleshooting your issues. So with that, I would like to jump into the data. So this presentation, I'll be primarily focusing on prostate cancer, how successfully we use this approach for studying prostate cancer. So like I mentioned, this is not limited only to prostate cancer. If anyone is interested, you'll be able to use for other cancer type as well. So as you know, that prostate cancer is the kind of well characterized, well studied at both at the genome and the expression level. And it was only the prostate cancer was the first kind of solid cancer which was identified to have some recurrent gene fusions like the TMPRSS2 gene fusions and other its family genes. And I was involved in the discovery of BRAF and other gene fusion, which I will show you in the next slides. So I'm going to use these important prostate cancer specific biomarkers, how we can actually study in depth for the expression pattern of these genes and how to understand the tumor molecular heterogeneity of prostate cancer. So as you know, these are some of the markers that are known to be kind of recurrently rearranged in prostate cancer, like the TMPRSS to ERG gene fusions, then the SPINK1 overexpression, PTEN deletion in about 30% of the prostate cancer, then ETV1, ETV4, and ETV5 gene fusions, and PCA3, PCAT1, and SLAP1. These are the well-characterized non-coding RNAs. Then recently we showed the recurrent gene fusion involving the KLKP1, which is a pseudogene that is forming a gene fusion as well. So these are the well-characterized prostate cancer specific molecular biomarkers. So either you can study these markers individually just by using by IHC or by RNA-ish if the antibody is not available, or you can actually detect more than one marker simultaneously. Okay, so here in this presentation, what I'm going to show is either you can actually visualize these markers individually or 3 or 4 markers simultaneously in the single hybridization. So I will show you how we are doing it. By doing that, we have a good understanding about the molecular heterogeneity of the prostate cancer. So the first, like ERG, PTEN, and SPINK1, there are good antibodies are available, and they are well characterized, and it is published in several papers. So you can easily perform IHC either individually or you can combine like ERG and PTEN. You can do because ERG is nuclear, PTEN is cytoplasmic, or ERG and SPINK1. ERG is nuclear and SPINK1 is cytoplasmic. So you can do it either as a single color IHC or as a dual IHC. That can be done. But for ETV1, ETV4, and ETV5, there is no good antibody. Okay, although there are many antibodies that are being sold by different vendors, but none of those antibodies are specific for cancer. If you do it on the tissue, you will see them staining all over. Okay, they are not cancer specific like the ERG antibody. So it may work well on the Western blot, but on the tissue, they are not good for in situ analysis. So that's why we developed the RNA-ish method for visualizing ETV1, 4, and 5. Then obviously the PCA3, PCAT, and SLAP, these are all, and the KLKP1, these are all non-coding RNAs, and the KLKP1 is a pseudogene. So these can be detected only by RNA. Yes, there is no, since these are all non-coding RNAs, there is no antibody available. And finally, what we did was we will be able to combine ERG, SPINK, ETV1, ETV4. Like you can do all the 4 markers simultaneously in a single hybridization. So by doing why we are doing this and what we learn out of this, that I will show you in the subsequent slide when we move to that those slides. So first, to begin with, since we spent a lot of time initially to set up this technology in our lab when the ACD Bio was new, that was in the 2000, I think 8, 9, or 10 in those days. The technology was not well perfected, or it was not quite popular. So we were one of the early adapters, and I thank ACD Bio technical team. In fact, some of them, they came to my lab and they helped me to set up this assay. So here I will show you how comparable the RNA in situ hybridization is comparable with the immunohistochemistry. Okay, I'm going to show you just one example. So here before, like I said before, before you do any of this RNA in situ hybridization, make sure you test those slides with the control probe. So here we did the Pol R2A as a control probe, make sure the RNA integrity is high, so that then use the positive and negative control to make sure they are working. So on the same tissue, what we did, so we took a case that is known positive for the ERG gene fusion. So this is the ERG immunohistochemistry that shows the nuclear staining only in the cancer, not in the stroma or in the benign tissue. So we took a consecutive slide from the same patient and we did the RNA in situ hybridization using the ERG specific probe. As you can see, the staining is specific highly specific only in the tumor area, like the IHC. You don't see anything in the stroma or in the benign. So it is yes or no. It is not like you're not looking for differential between the benign and the cancer. Okay, so I think that is the beauty of this ERG gene fusion expression, because these genes are expressed only after the chromosome translocation. So there is no endogenous level of ERG expression in the benign tissue. It's only when there is a chromosome translocation, the ERG gene is activated by androgen responsive elements and it gets overexpressed. So this is a good example to show that both RNA and the RNA and IHC is quite comparable, and you will be able to visualize the tumor without any ambiguity. Okay, so next one. So I think based on several kind of samples that we have analyzed using this RNA-ish method, so we came up with a kind of a scoring system, because you will see sometimes the genes are expressed at different level, whether it is a low or a high expression. So we have a kind of a scoring system. When it is negative, it's like 0. Or when it is when you're seeing the varying pattern based on the intensity, we give a different grade, like 1+, 2+, 3+. Because the beauty of RNA is you will be able to visualize individual mRNA transcript as like a distinct punctate dots. So every dot represents the messenger RNA molecule that we are seeing it. So depending on the level of expression, you will see the different intensity changes. So next I will show you how we use this RNA-ish for the detection of ERG gene fusions in prostate cancer. So as you know, I think with the discovery of the ERG family gene fusion in prostate cancer, this is for the first time we will be able to classify the prostate cancer into distinct molecular subtypes. So we have ERG fusions combined together, which account for about 50% of prostate cancer, then SPINK1 in about 10% of Caucasian and about 30% in African American. Then I was involved in the discovery of the RAF kinase gene fusion prostate cancer, which are present in about 1 to 2% of the prostate cancer. And recently, some of the non-coding RNA targets have been identified. So this, for the first time, we were able to classify the prostate cancer into distinct molecular subtypes. So the other interesting thing is these molecular markers or the gene fusions, they are not associated with any particular Gleason grade. Like ERG gene fusion, you will find even in high-grade PIN or Gleason 6, 7, 8 on all the stages, like the other markers also. So it is important that just by looking at the morphology alone is not helpful for to decide the risk factors or the prognosis of the disease. I think combining this molecular evaluation helps to assess the risk for recurrence of the disease and other things. So it is not only that these markers are important. Recent work, some of the preclinical work clearly show that there are some therapeutic drugs can be developed targeting this gene fusion. So some of these drugs, although they are not available for prime time clinical use, but they look promising. That some of the preclinical studies that clearly shows that they have a potential. Like for the RAF kinase gene fusions, and there is already sorafenib for the MEK inhibitors, so there are already FDA approved drugs are available, so that can be used as well. So given this importance of these markers, so we need to have a robust system for testing the tissue samples for the presence or absence of this gene fusion. So in that regard, I think we need to have a very reliable and a robust method for the detection of the gene fusions. Every time you don't have to do PCR or next-gen sequencing. We can have, even if we have a small biopsy tissue, that can be easily assessed by doing this IHC and the RNA-ish, which I will show you how we can do that. So these are some of the probes that are actually originally synthesized by ACD Bio. So for ERG, ETV1, ETV4, and ETV5, So these are the mRNA with all different exons in the number, and the bar below shows the location of the RNA-ish probe. Okay, so it is not that you cannot take the whole gene for making a probe. I think ACD Bio, they have the algorithm that they will be able to kind of synthesize a gene-specific probe, because the X gene, they have about 27 gene family members, so there will be some high sequence homology. So in order to avoid any cross-hybridization of the probes, they will carefully select the regions that will give a unique probe for each gene. So here, in order to evaluate, because since we were the one first doing this RNA-ish for these gene fusions, we did our due diligence to show that these probes are indeed, they are specific for each gene. Like in the top panel, like the ETV1 probe, so we will see only the ETV1 positive case is actually strongly positive, but the other, like when you do the ETV4 and ETV5 on the same case, they are not showing positive, because although these genes are kind of a family of genes, they have kind of related sequences. So we developed the probe that is specific for each gene. Okay, so that is the beauty. And in the bottom, you will see the ETV4 probe is actually positive only in the ETV4 positive case, but it is negative in the ETV1 positive and ETV5 positive cases. So that clearly shows the specificity of this probe. So having evaluated this probe for the specificity, then we did some systematic screening of prostate cancer. So if anyone wants to kind of screen the prostate cancer for the presence or absence of the gene fusions, so you have to go in a systematic way. First to the pathology specimen, whether it's a needle biopsy or the radical prostatectomy samples. First the pathologist will review for the presence or absence of cancer. So if it is benign, we don't have to do anything, but if it is a cancer, first depending on the size of the tumor, if there are atypical lesions or something like that, they will combined with the AMACR staining, just confirm it is cancer, or some cytokeratin, like basal cell cocktail, like P63 and the high molecular weight cytokeratin. So that will rule out whether it is benign or cancer. So once you rule out the specimen contains cancer, then you can do a systematic characterization of the gene fusions. Depending upon the frequency of these markers, since ERG is the most prevalent marker, first you can do the ERG-IHC or the ERG-FISH, you can do it, then followed by SPINK1, then you can do ETV1, ETV. So I think the main purpose of this slide is to show that if you want to screen all these markers individually, you'll be wasting a lot of slides, resources, time, and everything. Because sometimes when you have biopsy, you cannot afford that many slides as well. Okay, so most of the time the biopsy tissue, they may yield like not more than like 5 slides or something like that, because that will be used for several other. You have to do H&E, then other markers and everything. So given the limitations of the available tissues, so you don't want to do this marker screening individually. So it is important that you want to have a simultaneous evaluation of all these markers just by using a single slide. So I will show you how we did it. So this is an example to show the IHC and the RNA-ish for individual markers. So we selected the needle biopsy samples from the kind of respective positive cases by ERG. By doing IHC, we showed nice positive signal. Then the SPINK1 IHC, also the tumor positive, then ETV1 RNA-ish, and the ETV4 RNA-ish. As you can see, the quality of the signal. The other beauty is the RNA-ish, you are going to do only by bright field. Although you can do by fluorescence, but you can, it is easy for the pathologist and the lab people to visualize the signal when you are seeing under bright field. So this is the biggest advantage. So we have kind of standardized the probe performance for all these markers on individual sections. So here again, I will show you another example in a large kind of needle biopsy. there is a one small tumor foci that is positive for ETV1 to see how specifically, you can recognize the positivity. You don't see in the in the stromal area or the benign, you don't see any kind of background or non-specific staining. So the probes are very specific. They will pick up each and every tumor cell. So the advantage of doing this is, you can do some automated, by using the digital images, you can do some automated quantitative analysis of the signal to get an idea of what percentage of cells are having these positive markers. Okay, so this clearly shows the specificity. Because I'm showing all these things, we are not, we did not just do these assays on one type of tissue. We did on varieties of tissues. We did on the needle biopsy, and here I'm showing you on a tissue microarray. Here there are hundreds of samples here. By doing the ETV1 RNase, I clearly show that only there are only 3 cases are positive for ETV1, and the rest of the cases are distinctly negative. So this clearly shows the specificity of the probes. You can easily, reliably or categorically, you can pick up those positive cases without any issues. That shows the power of this approach. Then it is not only the TMA or needle biopsy. So this is a patient-derived xenograft tissue, which is a ETV1 positive tumor that was actually implanted into the mouse, and it is growing as a tumor. As you can see, only the human tissue that carrying the ETV1 rearrangement positive, it is showing strong positive, and then all the mouse tissue, it is not showing any staining at all. So it is not cross-hybridizing with the mouse gene ETV1 or anything like that. It shows the very highly specific probe that will pick up only the gene of your interest. So these are some of the examples to show the specificity of the probe. So we already published this work. If anyone is interested, please go through this paper. You'll be able to get more information about how we did the methods and everything, the procedure. So next, since we have optimized the conditions for the probe and we standardized the conditions for both IHC and RNase, so now I will show you how we identified some new molecular subset of prostate cancer by doing a dual color RNase and a dual color IHC. Because I think traditionally people do like only one marker at a time, then call it a day, it's positive or negative. Because the prostate cancer is highly heterogeneous, I think I will show you some good examples. You will appreciate what I'm talking about. So here, what I'm showing is we were evaluating one of the case that we know that is positive for ETV1. Okay, positive for initially we confirmed by FISH, because before we do that we don't have any RNase method or no antibody, so we confirmed by FISH and PCR that this case is positive for ETV1. So then when we did the ETV1 RNase, so all the red color staining is the ETV1 expression, as you can see, but there are 2 areas that is circled there. They are still cancer, but they are not positive for ETV1. So that really puzzled me. So initially, I was challenging my technicians. It could be some air bubbles on the slide. That's why the probe didn't reach that area. But I asked the technician to repeat again, and again we got the same result. So that gave me some idea what is really going on. So this clearly shows the tumor heterogeneity. One area is positive for ETV1, and there are areas that are negative. So out of curiosity, what I did the ERG immunohistochemistry on the same on another consecutive slide. So interestingly what I found was the area that is negative for ETV1, now they are actually positive for ERG. Okay, so because I selected ERG because ERG is the other prevalent marker. So when I did ERG, so that area is positive for ERG, and immediately area next to that was positive for ETV1. So this is the zoom in view of the ERG. And also we did FISH to confirm that these different foci in this tumor are positive. One is this is positive for ERG, and the other area is positive for ETV1. So this is the first time that we were able to show that you can have this what is called the intratumor heterogeneity. Like one area is positive for one marker, other area is positive for one marker. So if you kind of close the analysis just by doing only one marker, then you will be missing an important information. So that is the powerful approach by combining both IHC and RNA-ish. This was the first time we were able to show that the simultaneous evaluation of both ETV1 and ERG gene fusion on the same case. So here is another example. There is a small area high-grade PIM that is positive for ERG, and immediately right next to that, the adjacent area, it is positive for ETV1. There is an RNA, the very next foci. So there is no kind of overlap. So they are adjacent, but you will see the distinct the molecular feature. This foci is positive for ERG, and the one next to is positive for ETV1. So there are two different kind of mutation occurring in the same patient in two distinct foci. So this is the important information. For the first time, we were able to do it. And there is another case also, here also another example. There is a large area that is positive for ETV1, and at the bottom, there is a small area that is positive for ERG, but still there is a third foci, very at the bottom, still it is negative both for ERG and ETV1. So it could be something else going on, okay? So we don't know what it is going on because we screened the other marker, they are not positive. So it could be some new mutations that is yet to be discovered, or some new gene fusion is not. So this clearly tells the enormous the tumor heterogeneity, the inter and intra-tumor heterogeneity, which you can actually resolve by doing this combined IHC and RNA-ish. Okay, so that is the application of IHC and RNA-ish. The next, because so far we did this IHC and RNA-ish on the selected tumor foci. Like when you have a radical prostatectomy or needle biopsy, you know you are taking only one tumor foci and doing all the analysis. So now I will show you, without any kind of tumor selection bias, how you can actually understand this tumor molecular heterogeneity by doing this IHC and RNA-ish. So here I will show you, because I showed you we can do both needle biopsy and the radical prostatectomy and the tissue microarray. But what we know is more than about 80 to 90% of the prostate cancer are multifocal disease, meaning that there is more than one tumor foci that you will find in the prostate gland. Okay, so if you are doing the radical prostatectomy or needle biopsy, you will be able to sample only one tumor, right? And I think the prevailing thought is about you always look for the large tumor and high Gleason grade, and people thought that those are the ones that driving the progression of the disease. But now eventually that idea is kind of changing. Now people are looking at both the dominant or the index foci and also all the secondary foci, and they are giving a combined Gleason score like that. So for that reason, what we did, instead of kind of selecting a particular tumor foci, we wanted to evaluate the entire kind of the prostate without selecting any particular area. Okay, so here I will explain why we are doing that. Because as you know, the prostate cancer development is a kind of 10 or 20-year process. So initially, you will see some high-grade pain or something like that developing. Then slowly, you will see the tumors of different grades are existing within a prostate. Okay, so when you see this kind of multifocal, both from low-grade tumor to high-grade tumor, what is the relationship between this tumor within the prostate gland? When you have a multifocal, because like I said, there are 90% of the prostate are like that. Okay, you don't find a single tumor foci. Most of the time, you will find more than one tumor foci. In such situations, are these tumor foci are genetically identical? Or in other words, do they carry the same mutation or not? This is the fundamental question that we wanted to answer. So for that, this technology is really helpful. Because before I show that, I will show an example. So this is a study that people have done both by FISH and next generation sequencing by selecting tumors from different locations of the prostate, and they did some extensive sequencing, and they are able to kind of generate this phylogenetic tree where there will be some commonality in all the tumors in terms of aberrations, but as the tumor progresses, you will see subclonal population. They will carry some unique aberrations like that. So this is how the tumors are branching out. It starts from one, and you will see that. So when you do this type of analysis by doing the next-gen sequencing or everything, you are going to just crush the tumor, take everything, and you are going to give a more kind of a global analysis. But that is not going to really helpful. Okay, so what we did was we just preserved the tissue as it is. Okay, so first this is a study that we published. Initially, what we did here, we took some needle biopsy samples. So in a needle biopsy, you will see in the long core, one end there will be a tumor foci, in the other end there will be a tumor foci, in between is a stroma or benign tissue. So when you see this kind of two tumor foci, we ask the question to whether these two tumor foci are the same. So what we found, there are more than about 29 or 30% of these what is called discontinuous foci, what we call it. Okay, discontinuous means these tumor foci are not connected, because either we don't know whether it is like in the figure A and B, as you can see, whether they are coming from the same foci or from two different foci when you take the needle biopsy. So that's the reason we wanted to do this study. What we found was there are more than about 30% of these discontinuous needle biopsy specimens carry, like one foci is positive for ERG, other foci is positive for ERG, other foci for SPINK1, or one is negative or other one is positive, or one marker like that. So we clearly demonstrated this type of heterogeneity using the needle biopsy specimen. So then we extended the study. So here is another example to show the discontinuous foci. So you have the in between, the benign, at the top there is a ETV1, and the bottom the another tumor foci that is ERG. That clearly shows, because when you look at the Gleason grade, they both are kind of identical. You may like call it Gleason 6 like that, but at the molecular level they are completely different. So this is a very important information that we are learning by doing both IHC and RNase. Suppose if you do only one marker, since ERG is present in 50% of the cancer, here you are going to see only one foci that is positive for ERG, the other foci you call it negative, you don't know what is going on. So just by doing both IHC and RNase, we were able to show this intertumor heterogeneity. So given this type of kind of tumor heterogeneity in the discontinuous foci, what we did, so evaluation of just one marker or two marker is not sufficient. Since we know that ERG is there, SPINK1, ETV1, ETV4, these account for about 60% of the prostate cancer. What we did was we want to combine all the four in one experiment. Okay, so that we want to analyze the simultaneous expression of all these four, if at all they are expressed. Okay, so what we did was, as I told you before, the ERG and SPINK1, we have a good antibody, so we developed the dual IHC. So the ERG will be detected with the green chromogen, and the SPINK1 with the blue chromogen, and the ETV1 you can do it in brown, and ETV4 in red color. This is RNA-ish. So you can do both RNA-ish and IHC on the same slide. Okay, but of course you will be doing in a sequential manner. You cannot do both IHC and RNAs at the same time. Like I said before, first you do the RNA-ish, then you do the IHC. But RNA is either you can do it as a single probe or a dual probe. Okay, this is what we did. So here clearly I'm showing an example. So this is a tissue microarray that we constructed, putting all the positive cases like ERG positive, SPINK positive, ETV1 positive, ETV4 positive, then some fusion negative cases. So this is a TMA that we constructed purposely for this study. So when we did both IHC and RNase, that is 2-marker IHC and 2-marker RNase, we were able to show on the same in a single slide all the 4 markers. Like in the top, that is an ETV4 positive tumor. Then the next to that is a SPINK1 positive tumor, and in the bottom, in the middle, you have the ERG positive tumor, and in the bottom, you have the ETV1 positive tumor. So this is the beauty. The one good thing of all these markers is if a tumor is positive, one tumor, if it is positive for a particular mutation, you don't see a second gene fusion in the same foci. It may be in a different foci on the same prostate gland, but not within the foci, you don't see a second mutation. So these are all kind of mutually exclusive, these markers, that's how they express. So it is easy for us to detect. And other important thing is, as I told you before, since these genes are expressed only when they undergo chromosome translocations, so there is no endogenous level of expression of this. They don't express in benign at all. So you can clearly see the differential. We are not looking for a differential expression of these markers. But like I said before, if you are working on other solid cancers, if you are looking for a differential expression like low in benign or high in tumor, or low in tumor or high in benign, like that. So when there is a differential, there you can easily quantitate as well. So that is not really challenging. So what we did, that is not only just that TMA. So this is the another large TMA. Okay. Because that TMA was constructed for that purpose, but this is an entirely new TMA that we got from our collaborator, and we did this 4-color IHC and RNA-ish, and we were able to identify these distinct positive cases, where I will show you a little zoom-in view. See this? This is the ERK positive cases. The ERK is positive only in the tumor, and this is the SPINK one. That is the cytoplasmic staining. Then this is the ETV one again. Because since we are doing, so you may be wondering why you are seeing this green color in between, right? So these greens are basically the ERK antibody. It is not only staining the prostate tumor, but it's also expressed in the endothelial cells. So that serves as an internal control for this antibody. So that's why even in the, since we did both IHC and RNAs on the same slide, so even in the, after we did, even though this tumor is positive for ETV1, you will see some ERK staining in between on the endothelial cells as well, and similarly for ETV4 as well. Okay, so that is the reason you will see some green spots. So when the tumor is negative, clearly for ERK, ETV1, ETV4 is completely negative, and only you see the endothelial staining of the ERK, only the green. So this is a very perfect example of how specific these antibodies and the RNase probes are. You know, they detect only the positive cases, and you are not dealing with any false positive or false negative cases. Okay. So now using this approach, so like I showed you before, either you can do it on needle biopsy or radical prostatectomy sample using a selected tumor or a tissue microarray, because those are all heavily biased. You are selecting some tumor of interest. Okay. So given this enormous heterogeneity in a multifocal cancer, what we did, we don't want to do any kind of tumor selection bias. All right. So we want to evaluate the tumor heterogeneity in situ as it is by using the whole mount radical prostatectomy specimens, meaning that the entire prostate is actually fixed in a block by using a 3x2 slide. We will be able to section the prostate gland and even put it on a large glass slide. This is not the standard microscope slide. So this is the double the size of the standard microscope slide. It's a large microscope slide. You'll be able to mount the entire prostate on a glass slide. Then you can do this in situ analysis. So here I'm showing this example. So this is the entire prostate, and the gap in the middle is the urethra and all those things. So in one area, you will see there is a tumor here that is positive for ERK. Then other area in this large square, there is a tumor. But interestingly, what we found was when you look at the morphology or the H&E, they all look the same, like a Gleason 6, something like that. But when we did this IHC and RNA-ish on the same slide, what we found was one portion of this section on the left side is completely positive for ERK, and on the right side is positive for ETV1. So this clearly shows that these are two different tumors, and they are colliding tumors. Okay. So they are evolved independently. Since they are expanding, they look like they are merging together. Under H&E, they are going to look like one tumor. But when we did this molecular analysis, we were able to separate, okay, these tumors into distinct molecular subtypes. So that is the power of this approach. You will be able to evaluate the tumor heterogeneity by using these known cancer-specific markers. The next one, this is another interesting case where there are 5 tumors. In which the large index foci is at the bottom, which is completely negative, But when we did the ERG, ETB1, and SPINK1, three markers, what we found was like one tumor is positive for ERG, other one is positive for SPINK1, then the third one is positive for ETB1. Okay, so there are three markers that are present in the one prostate. Okay, so this is the very powerful kind of information that we were able to get for prostate. This is the first time we clearly show this enormous tumor heterogeneity. Suppose, if you are going to make clinical decision just by looking at only the dominant or the index foci, you are going to completely miss all these positive tumor foci here. Okay, so the patient is not going to get the real benefit. Suppose if there is a drug or something is available for ERG or SPINK1, something like that, so you are not going to treat this patient. You are going to get completely a false and negative result based on the analysis of the index foci by missing all the ERG, SPINK1, and ETB1 like that. So this is the first time we clearly show this extent of this tumor molecular heterogeneity in a multifocal tumor. So I'm sure you are convinced that how powerful this combined IHC and RNase approach is useful to studying the tumor heterogeneity. So this is not just for prostate cancer. If anyone is interested, if you have different markers, you can develop this approach for other cancer as well. I think already people are working on the PD1 markers, they are showing heterogeneity, then the HER2 markers, So you can combine with additional markers to show what exactly is going on. So this is the, because although the ERG and ETB1 are the same X family gene fusions, but what we found was, based on the recent literature, they function at the molecular level entirely differently. Okay, so the ERG and ETB1, they control a common transcriptional network by largely in an opposing fashion. Okay, so although they are same gene family, but they are functionally, they are totally different. Like ERG is negatively regulating the androgen receptor transcriptional program, but whereas ETB1, it actually cooperates with AR signaling by favoring activation of AR transcriptional program. These are some of the papers that are published. So then also people have shown that ETB1 expression, but not the ERG, promotes autonomous testosterone production. And the ETB1, but not ERG, upregulates expression of AR target genes as well as genes involved in steroid biosynthesis and metabolism. So ETB1 supports the development of invasive adenocarcinoma under the background of full PTEN loss. So like that, there is a distinct biology that are associated. So in the prostate, one tumor foci has ETB1, other is ERG1, and they are functioning in an opposite way. They make the cancer is much more aggressive, or the patient may have a more aggressive disease or a bad prognosis like that. So evaluating these tumors, although we don't have drug, there is no direct clinical use for these markers, but I think using these markers, by studying this heterogeneity, we will be able to understand the outcome or the risk factors for the prostate in a much better way. I think for that reason, when we combine this, both IHC and RNase, that will be a powerful approach. Since we can do both on the needle biopsy and on the whole mode specimens, we will have a better understanding of the tumor prognosis. So next, quickly, I will show you how we can do RNase for non-coding RNA. As you know that there are many non-coding RNAs recently emerging as a potential kind of biomarkers for cancer. Like several, they have some known functions. They can function as a kind of gene regulator molecules. They can work as a protein inhibition or the post-transcriptional modifiers or decoy elements, like that, So that's why, since we don't have an antibody, or we cannot, since they are not coding for any protein, so if you want to evaluate the tissue level expression of non-coding RNAs, I think RNase is the best method for evaluation of the expression of this non-coding RNA. So here I'm showing an example of PCA3, which is a prostate-specific non-coding RNA that is known to be highly expressed in about 90 or 95% of the cases. And there is a urine test that is FDA approved. It's called a Progensa. There is a urine test is available that is FDA approved, and the people are using it, combining with the ERG and PCA3 and the PSA level. They can actually predict for the presence of cancer by non-invasive methods, because TMPRSS to ERG you can detect in the urine, And PCA3 also, you can do it in the urine, then you can do the serum PSA. So everything without even taking the biopsy, by combining both the ERG, PSA, and PCA3, you can come up with a score, and that will predict whether you are going to see a cancer when you do the biopsy or rebiopsy like that. So even when the biopsy is negative for the first time, when you do this type of analysis, because one marker is not absolute, because these markers are representing only a small percentage of the cases, like ERG is present only in 50%, PCA3 more than 90% of cases, then PSA is always elevated in the tumor cases. By combining all these three, you will be able to come up with a reliable diagnosis whether the patient has advanced cancer or not. Okay, so in that reason, so we did a study showing whether the urine, since PCA3 can be evaluated in the urine, so we did a study assessing whether there is a correlation between the tissue level expression of PCA3 and the same cases matching urine as well. So there is a good concordance there. So I think given the time, I will quickly, I don't have time to go through that on the PCA3. So like I said, the PCA3 is a good marker. More than 90% of the prostate has this marker. There is a Progensa urine test is available. So what we did was, for the first time, by using the RNA-ish, because before our work, people have used some radioactive probe for the PCA3 and they showed the tissue level expression. But we were the one first, for the first time, by using the ACD Bio RNA-ish probe, we showed that the expression of the PCA3 either in a kind of small area of the prostate, or you will see the entire prostate may be positive for the PCA3. As you can see, almost every tumor area in this sample is positive for PCA3. Okay, so this is how we can actually evaluate the tissue level expression of the PCA. I think this for the first time, we showed the tissue level expression. So we published this paper. If anyone is interested, please go through this paper. Here we show the concordance of PCA3 expression both in the tissue and the urine level. This is the first time that we showed there. And the next I will quickly show you the DNA and RNA, DNA fish and RNA-ish on the same slide. Okay, here just one example. So the DNA and I think the green color signals are using a DNA probe for a particular gene of our interest. Then we use the RNA probe. So on the right side, you will see those pink dots where the RNA is expressed. So in the same cell, so our goal was, since if we use the DNA probe, whether the RNA is also expressed, because we know that this RNA, this is more like it has a function of a DNA binding for gene regulation. So the main purpose of this experiment to see whether the gene locus identified by the DNA fish, and we asked the question whether the RNA is functioning at the same foci, same locus, or in a different locus. What we found was the DNA copy number is intact, but the RNA is actually functioning in a different location in the DNA, in the genome, like that. So what I'm showing is here you can do both the DNA fish and the RNA fish on the same slide to evaluate the status of the DNA copy number and the RNA level for the gene of interest. So this is a very powerful approach that anyone can use it. And it is not only, like I said, it's not just for prostate cancer or anything like that. So we published a paper for some of the sarcomas where we have some of the known genes like the X family genes are also rearranged in the sarcomas. So we did some study on the sarcomas as well. And by using the RNA-ish, we were able to detect some of the virus as well. This is called a Merkel cell carcinoma samples. We were able to show the positive for some viral probe that we used, and we were able to show it. So the application is broad, by using the RNA-ish, and if necessary, you can combine with IHC as well. Okay, so this is the power of both IHC and RNA-ish. So in summary, I showed you an overview of what are the applications of RNA-ish and the IHC, how you can combine both IHC and RNA-ish for the simultaneous evaluation of up to 4 markers. I think now, I think this is the limit of this technology right now. I'm sure the technology is evolving. they will be able to, because the limitation is the availability of the chromogens. Okay, if we have more distinct chromogens are available, we will be able to add more markers. I'm sure the company is working towards that to making it more multiplex. So at this point, I clearly show the application of combined IHC and RNase, how you can actually detect the S gene fusions in prostate cancer, and we identified new subset of prostate cancer where more than one gene fusion present in the same tumor foci. And then also I showed you that we can analyze different type of samples, needle biopsy, radical prostatectomy, tissue microarray, and patient-derived xenograft tissues. You can use any type of tissues. In fact, I forgot to use here, if you have a fresh frozen tissue, we can do RNA-ish on the fresh frozen tissue as well. So there is no limitation. Okay, so once it is fixed in formalin, the fresh frozen tissue, or if you have a touch prep, I think that is something that the pathologist or the clinician, they may want to get some quick results. If they want to see the results, if you have a touch prep specimens, you can do RNA-ish on that as well. Because RNA-ish procedure is you will get the results on the same day. If you start in the morning, by early afternoon, you will be able to get the results. You don't have to, there is no overnight incubation or anything like that. If you have the slides, start in the morning, and by evening you will get it by manual method, and also I think automated method is like less than 5 hours or something like that. So on the same day, you will be able to get the results. So this is a very powerful kind of technology that can be widely used in all different type of tissues. I hope I convinced you by showing the application of combined IH and the RNase. So with that, I would like to thank the entire team of clinicians, pathologists, and the lab people who have been very helpful in setting up this technology. And I think recently we have published several papers. You can search with my last name, and you will be able to find some important papers that we published both in Modern Pathology and the Clinical Cancer Research and other journals as well. So with that, I would like to thank you for your attention, and I will be happy to answer any questions. Thank you very much.
Thank you very much, Dr. Palasamy. That was an excellent presentation. Really appreciate you taking the time to go through in so much detail. That was really wonderful. Here's an opportunity, if you have not yet asked any questions, if you have any questions for Dr. Nala, please do pop them into the chat. In the meantime, I do have a couple of questions lined up for you that we can go ahead with. Dr. Nala, what are the advantages of using RNA in situ hybridization over IHC?
Yes, as I mentioned in my presentation, RNA is not a technique to replace IHC. RNA is a unique and technology that can be used for the genes of interest for which you don't have a good antibody. So even though there are some antibodies available for some of the genes, like different vectors have different antibodies, and they may have a different kind of sensitivity and specificity, and they may have some cross-reactivity and all those issues. In those situations, RNA is the best method to overcome all those issues, because RNA is a sequence-based hybridization. So you will not get any non-specific or off-target signals. The signals will be very specific, and also the way the RNA works is actually, it will hybridize only to the gene of interest, and you do not see any off-target or non-specific signals. So it is for that reason, for the genes for which even if the antibody is available, if the different antibody performance are quite variable, you can actually cross-validate your IHC data using RNase. That is one application. And for genes for which if you don't have any antibody, and if you are interested in studying the expression of this gene at the tissue level, RNA is the method of choice for the protein coding genes as well. And like I said, now the non-coding RNAs and the pseudogenes, these are the new molecular markers that are evolving, and their role in cancer is well established. So in order to study the tissue level expression of these non-coding RNA molecules, RNA is the only option right now for the in-situ detection, although you can do PCR, next-gen sequencing, and all those things. Because as I showed you, because of this enormous tissue level inter and intra-tumor heterogeneity, if you want to see where these molecules are expressed within a tumor, because you are not expecting them to express in all the tissues, if you want to see the tissue specific, or then you want to combine with the morphological features, I think RNA is the best method. The other main advantage is, you can do RNAs under the bright field method. Okay, so that is the beauty of it. Like IHC, you can visualize the results under bright field. Of course, there is an option to do fluorescence as well, but I think most of the time, I think bright field is the best approach, because you are preserving the tissue morphology. You can have the counter stain like a H&E, so you can relate of your expression results with the tissue morphology, what type of tissue morphology that you're seeing. So I think these are some of the advantages of using RNA-ish over IHC. So each one has its unique advantages. I'm not saying that RNA is replacing IHC. You can still have IHC, where as long as you have a fully validated and a highly specific antibody, that can be still used. Then in place, if you want to study additional markers, you can put RNAs for that. And in that reason, I think both techniques are complementary, but RNA is really kind of sequence specific, and I think the sensitivity and specificity are much higher. For that reason, I recommend using RNAs if you have issues with any of the antibody that is available for any of the known genes. Yeah, thank you for the question.
Great, thank you very much. That was really good. Okay, I do have another one here. Can RNA-ish be performed along with DNA FISH?
Yes, like I showed you in the last slide, I think that is the other important development in this field, because FISH, as you know, that is a quite popular method, and people are using it for detecting DNA amplifications, deletions, translocations, and small inserts and deletions and all those things. So when you do just the DNA, you will get only limited information. Suppose if you're dealing with an amplification of a particular gene, although it doesn't necessarily mean that when a DNA level copy number is increased, it doesn't necessarily mean that the gene within that amplicon is also highly expressed at the RNA level. So in order to answer that question, if you have a small focal amplicon where there are 4 or 5 genes within the amplicon, for example, so if you want to know what is the expression pattern of the genes involved within the amplicon, so in that situation, you will be able to have a multicolor DNA FISH. So like, the DNA FISH can be done with the 3 or 4 colors like that. You can do multicolor DNA FISH. Then on the same slide, you can do the RNA-ish, because RNA signal is going to be look entirely different, like the DNA FISH, and you are capturing in different layers. So you will be able to simultaneously visualize both the DNA copy number and the RNA expression. So that is the power of this. So I think that is the new development of this. Originally, RNA was developed as such, but there are new associated development that is coming up with that. So yes, to answer the questions, we can do both DNA FISH and RNA-ish. Thank you.
Great, thank you very much. And yes, we do have a number of new items in the pipeline, so do watch this space for more information on that. Thank you very much, Dr. Nalla. That was an excellent overview of using ISH and IHC together, combined. I really appreciate you taking the time to speak today. Thank you.
Thank you very much for the opportunity. Thank you all. Please email me if you have any questions. I'll be happy to answer, or I'll be happy to help you if you are setting up this RNase procedure in your lab. Either you can contact me or the technical support from the ACD Bio company. Thank you.
Thank you. That's wonderful. And if we weren't able to get to your questions offline, please do continue to pop them in the chat, and we'll be reaching out to Dr. Nalla independently of today's session, and we'll make sure that we connect you and you get the answers that you need. Thank you very much.